Good morning, everybody. Welcome. People will still, I'm sure, what's the word, stream in. Traffic in Los Angeles is the usual. So anyway, it's really with great pleasure that I introduce as part of our once a month Autism Distinguished Lecture Series, Jacob Ellegood, who is a research associate at the Mouse Imaging Center at Sick Children's Hospital in Toronto. He got a PhD. Well, his bachelor's degree was in physics originally in 2002, and then he got a PhD in biomedical engineering from the University of Alberta in Edmonton. And those of you who know that area know that it's in the kind of the northern, most beautiful part of Canada, close to skiing, etc., but very cold. And so he's wearing his winter clothing. And he's really done extraordinarily focused and important work for those who are working in autism, but just animal models in general of neurodevelopmental disorders, in developing the kind of mouse MRI technology. But more than that is the kind of scope of the study, of the major study that he did, which really had a huge, it was a very highly cited paper. I think it was over 25, 26 something models of autism. So you have these rare Mendelian models of neurodevelopmental disorders that increase risk for autism. And he did a kind of structural imaging study and really, really careful analysis of that. And it kind of begins to, start to unravel molecular pathways connecting, genes to neuroanatomy, what the kind of developmental consequences are of these types of mutations. So he continues to do that. He's a major, and sought after who's doing mouse models, including us, and really has done spectacular work. So I haven't heard him talk maybe since Society for Neuroscience or something. Maybe possibly. It was. So I'm really looking forward to where things are right now.
Sure.
Thanks for taking all the effort to travel here.
Thank you very much for inviting me, and thanks for the nice introduction. As Dan mentioned, a lot of my work is looking at animal models of autism, and we try to collaborate with many people as that you remember blossomed into working on it. So I will be talking about my work on, now we're calling it animal population imaging, and looking at how, again, molecular pathways can link to the mouse, autism clusters in the mouse using neuroanatomy. Just a little background center. This is the new research at the Sick Kids Hospital, where they say everyone is now housed under the same roof, except for us, because we're over here. So the bottom two levels have tens of thousands of mice in it. And so Mark Henkelman, my old supervisor, used to say democracy in that place because we would be losing to the mice. In that facility, being that it's the mouse imaging center, a lot of what we do is image the mouse in a variety of different ways. We have a CT scanner, we have optical projection tomography, we have this serial 2-photon sectioner to look at cellular level differences in the mouse, but my focus has always been on the MRI. So currently we have two 7 Tesla MRI systems. This one is much, much older and possibly will be replaced in 2021. We do have funding for a new magnet, a new 11.7 Tesla magnet. But I think 2021 might be a little hopeful even at this point. But we hope this one lasts until that time, because that's where the majority of my work is being done on. This is our newer system. You can see that there's 4 coils in the center there for in vivo imaging of mice. So we do 4 at a time in our current system. And with that said, one of the important work and one of the benefits of working at the mouse their approach to high throughput multiple mouse imaging. That us to do these kind of projects where we can collaborate with 26 different labs and get their mice and scan 20 mice per group in a reasonable time frame. A lot of other mouse labs around the world are scanning one mouse at a time. 16. 16 is possible. Live, we tend to do about 7 or 4 on the new system, but overnight here, this is our ex vivo. So you see a little mouse brain there left in the skull, and there's 4 there, and there's 4 rows of 4. So we scan 16 ex vivo samples in one overnight session to get data for almost one of those collaborations in one overnight. This is the setup for I think they ran 16 live mice one time just to prove that they could do it, but they tend to, those who know about fMRI, it's most homogeneous in the center, and on the outer edges, the gradients are a little bent, so you don't get as great images. So we tend to just use the inner 7 of this hexagon. And that's just a little diagram showing you how the mouse is set up. He has a little molded sled and all the EEG and isoflurane and that stuff is all in there to anesthetize them while they're in the magnet. Example of some of our images. These are live images. They are 90 micron isotropic. For those that don't work with mice, this is the front, this is the back. Coronal slice up here, horizontal, sagittal. So this is actually 6 of the 7 coils shown here. So it's quite high detailed. These are where we now get 75 micron isotropic images, and we get these images in about an hour and a half on the scanner. So the mice aren't even anesthetized for very long. And again, these are manganese enhanced. This comes about because our new scanner has cryo coils, which cryogenically cools the coils, which increases the signal noise and therefore allows us better resolution and faster. The bread and butter of my work has been the ex vivo imaging, and this is 40 micron isotropic images, and so again, we're not down at cellular resolution, but we're getting closer and closer as we can. So we're talking mesoscopic resolution, which is in between microscopic and macroscopic. So you're looking at changes in regions more than changes in cell when I talk about the volume changes. This takes 14 hours to do. So that's why we want to scan 16 at once and why we do it overnight. So someone sets up the scanner at 4 o'clock or 5 o'clock, and then we come back in the morning and all our data is collected. That was true until that magnet broke down in June, and so it's been out of commission for 6 months. So we now have a backlog of about 500 brains. But we're hoping this month it gets fixed. Fingers crossed. And once we have all those images, we created this manually delineated atlas that highlights 182 different structures, and it's been organized in a hierarchical tree so that we can talk about all the different regions in the brain and say how their volumes change, either large-scale regions or the smaller structures on the inside. And currently, I have started scanning rats because there seems to be a push for genetically modified rats now than mice. But rats are predators for mice, so we always do them as ex vivo only because we can't house them in the mouse house. Yeah.
Yeah, could I just ask one question about the atlas? Just does that overlap with like the Allen Brain Atlas, other kind of molecular atlases?
It is. So the question was, I just have to repeat it, but the question was, Atlas overlaps with the Allen Brain Atlas, and it does. The delineations that are used in the hierarchical tree have been mapped directly to the names of what the Allen Brain Atlas is, so you can look at the gene expression and compare it directly to the region that we use. So for those that don't know, that is an adult mouse on top there, left in the skull, so it's not the brain. You're not looking at the brain, you're looking at the skull. And this is a 3-week-old rat. So there is quite a large-scale difference. So we actually, for scanning, we take our in vivo, our live coils, and 3D print it up a little holder and put the brain in that and scan it in our live coils because they have the size that is required for the larger brain. And we do this in about 3 hours and can get about 79 micron isotropic on the rat. So that's been a push from our collaborators like Jill UC Davis and Eric Morrow and Marcus Warr in Germany have started to send us rats of different versions. different genetic mutations. But with all that said and all that bragging about how good our imaging is, why I'm here is to talk about autism. When I started at the Mouse Center, that was 12 years ago, and Mark Henkelman, my postdoc supervisor, asked me to come up with an idea that used the mouse and used genetics and solve some kind of problem. And being that I worked at the Hospital for Sick Children, I thought of childhood diseases and thought we should see what we can do with autism. Naively, I thought that we would just be able to get the autistic mouse model and scan it and see how the brain was different, and then we'd have a paper and we'd solve everything. We even naively entitled our first grant that got funded through an imaging package, which is, it was where's autism located in the brain, which we haven't really solved necessarily at this point. A little history about autism. In the 1900s, there was no autism. There was autism, but it wasn't known. It was thought to be in the schizophrenia category. And then with DSM-2, this was after Leo Kanner had started the definition of autism, but it was thought to be mostly because of refrigerator mothers, so there wasn't an overall thought that there was a cognitive disorder that needed to be classified. So childhood schizophrenia tended to be how it was classified then. It wasn't until DSM-3 that it started to come out as infantile autism, and then childhood onset schizophrenia, and then again adult schizophrenia. And then it stayed that way for a while until I think DSM-4, where it split into a bunch of different categories. You had autism, Asperger's, childhood dyscognitive disorder, and but these kind of came under this pervasive developmental disorders umbrella. But I think sometime between DSM-4 and DSM-5, they figured out that there's too many genetics involved here that aren't putting these groups into these different categories. And they didn't go back, but they homogenized the diagnosis for autism into DSM-5, which just lists ASD. They took Rett's out, but a larger ASD group. Now, I don't expect you to read all this, and I'm sure you all know it, but the current definition of autism, basically social communication and social interaction deficits and some repetitive restrictive behavior. It's basically you need those two categories to have autism now. But with all of that said, yes, we have a nice definition. Yes, we have all of these things, but there is such a wide heterogeneity in autism that causes a lot of problems for diagnosis and treatment and everything. There's the famous, or not, I don't know if it's famous, but the quote I always say with autism, person with as everyone's different. So there's behavioral heterogeneity, there's, as Dan knows, quite a genetic heterogeneity, as well as what we look at as the neuroanatomical heterogeneity. And so behaviorally, these symptoms of these two hallmark characteristics are range from mild to severe. 40% tend to have above average intelligence, 20% are nonverbal, 30 to 80% have ADHD, 20% have OCD, so there's an overlapping comorbid disorders as well as a wide range in what an autistic person tends to look like. 70% of actually all patients tend to have another psychiatric disorder as well, so it is overlapping a bigger umbrella. Genetic heterogeneity, right now Safari gene database is going under a little bit of rework. It used to have over 1,000 genes, but they took out 2 of their categories. So now they list 800, and they're stronger candidates, so that's why they decreased it. No single gene accounts for more than 1 to 2% of cases, and you have ASD-related syndromes, which account for 10%, rare chromosome abnormalities for 5%, rare CNVs, or penetrant genes. But all in all, we get about, from genetics, we get about 25% of the reasons for autism in the population. It might change a little depending on who you talk to, but there is a large section of autism where there is an unknown genetic cause. So whether it's epigenetics or a gene by environment interaction, remains to be seen, but our hope is maybe we can figure some of that out. They have started to look at these genes and cluster them into different groups. So you have maybe the synaptic adhesion component genes, some that are transcriptional regulation, chromatin remodelers, or RNA processing. So they've tried to take these genes, these 800 genes, and categorize them to get a better handle on what autism looks like. That's one approach. There's also heterogeneity in autism with the neuroanatomy, which is where we like to look at. I always look at the literature and see, well, the hippocampus in autism has been revealed enlarged bilaterally. It's also been revealed to be significantly reduced, as well as no group differences. So there is a wide range in neuroanatomy, and I've used this slide for a very long time, and they're all taken from the early 2000s, late 90s. So I wanted to look at one of the most common findings in autism, the corpus callosum, and see if stuff has changed more recently. And it's the same story. No difference in the corpus callosum, reduced or significantly increased. So there is still this range in neuroanatomy, which makes it hard to find an autism signature, and why I can't say after the grant ran out where autism is located. So this heterogeneity causes problems. Treating a heterogeneous group can be difficult. What if it only works in a subset? That's going to ruin your primary outcome of your clinical trial if it's only going to work in a subset of the autistic patients. So what do you treat for? There's multiple brain differences, several possible mechanisms. So should all these autistic patients be in the same group and treated the same way? And the question I like to ask is, if we were to define autism now, given all that we know, would it still be called the same thing? Would it still be called autism and have the entire group? I'm not going to answer that, but it's just an interesting question to think about. So if we go back to this Sankey diagram, you have the DSM-5 definition. So where do we go from here is the question. You have the possibility to do it genetically. You have 800 different genes that are associated with autism. So you could divide autism into 800 different groups with all the different genes. I think that's not the way to necessarily go. That's going to be a lot of different small disorders of less than 1% of the population, and it's not going to be conducive to any treatment effects. You can take those groups and put them into sort of a transcription, adhesion, RNA gene, and that might get you a little further. But then you also have to factor in that we have to also bring in ADHD, we have to bring in OCD with autism, and maybe come up with this whole neurodevelopmental disorders group instead of just an autistic group. And then maybe we split that into 2 different disorders, or maybe it ends up being 10 different disorders. I don't know what the answer is at this point, but that's what we're working towards, getting better diagnostic specificity for autism to do it. But how do you explore a disorder with that much heterogeneity? Well, lucky for me, you can look at the mouse. The benefit of the mouse is you can control the environment. They all can be in the same environment, and you can control the genetics. You can compare basically an identical twins is what a C57 black sixmouth is supposed to be compared to its other litter mates. So you can control the genetics quite easily. The problem is there's 100+ mouse models of autism out there, just like, and now it's probably going to get closer to 800 by one for every gene. So where do we start with this? As Dan mentioned, we do as many collaborations as we can. We acquire as many mouse models as we can that are related to autism and scan them. Our current numbers, we've ballooned from the 26 after that paper came out, and now we've looked at current numbers are 125 different mouse models of autism, which is about over 3,500 MRI scans and greater than 10 years of work. And this is just showing the number of mice scanned from when I started in 2008 that were related to autism to 2018, where we get up to about the 3,500 group. So that's why we have to scan 16 every night, and why we need our scanner to be working again. So what do these collaborations usually look like? A mouse model, as we defined it for this, is it had to be genetically modified to resemble and or be related to a genetic lesion in human autism. Because of the way the literature is, we also included behaviorally autistic mice. So I'm talking the BTBR, it's somewhat controversial, but we wanted to include it as a model in this group as well. So we need about 10 to 12 mice per group to get accurate, to measure accurate differences between them. That gets about a 3 to 5% difference in volume can be picked up. So in some cases also we get 2 or 3 models per genetic lesion. So if I get a Neurexin 1 alpha mouse, there will be a heterozygous as well as a homozygous version of it. So I would count those as 2 different models. So in that 125, some of them would be both Neurexin. There would be 2 Neurexins in there. Each individual group will have its own wild type. There are significant differences in C57 Black 6 that are raised in my facility or Columbia, the University of Columbia, or Vanderbilt, and they change over time. So litter mates are best, but you need at least their own wild type from the same colony that the genetic mutant came from. Usually we try to get adults 60 days old. That's not possible in all cases because everybody has their own age that they're looking at. So we tried, but it's not always successful. And this is the paper that Dan was talking about in his introduction, where we looked at 26 different models, and with a bunch of different collaborators, 34 names on there, in order to get a better handle on this neuroanatomical heterogeneity. And this is what I've been building on. So we looked at, we arranged autism into 3 different clusters based solely on the neuroanatomy in that paper. We've expanded on that. This is the current listing of models. So this was the original group that was in that 26 model paper. And then we collaborated with the IMPC, the International Mouse Phenotyping Consortium, that is trying to actually knock out every single gene and create a mouse based on that. The Toronto Centre for Phenogenomics and the CMMR are creating some of the mice for that project. And so we got a list from Steve Schur, who's our genetics person at SickKids, and he gave us his in-house list, and we gave that to MR and said, who was on this list, and the ones they were able to get a hold of, they made in-house. So all of these ones were made in our lab for scanning. And then there's a bunch more for collaborations, and in some cases, because CHD8 was so hot in the autism community, we got it from 3 different labs as well as created one ourselves. In total, again, 125 were used. There's not 125 listed on this. Some are still secret until they publish. It's just the way it works. It's all in the data, but I'm not putting names on things. So have to trust me. You can ask me after, and we can talk about when I'm not mic'd up. So what do we do when we get them? Well, you saw the images from the MRI scan. So some of this information is just there for the people that know MRI in the audience. It's T2 weighted 3D fast spin echoes that we scanned 16 in parallel. These are some of the parameters listed there. And again, 40 micron isotropic, and it takes 14 hours. And this is one mouse. This isn't an average or anything like that. Quite nice imaging. Once we image him, for the original paper, we looked at regions, but for this, we had to look at voxels to not have any size bias in our comparisons. So what we use a technique called deformation-based morphometry once we have scanned the brains. Now what this does is let's say you have a mouse brain that has a large cortex and a mouse brain that has a thin cortex. You align them to create a consensus average. So the one with the large cortex will have to get a little smaller, the one with the smaller, thinner cortex will have to get a little bigger, and then it'll go into this average. And then let's say you superimpose a grid on top of that, and then work those deformations that were created backwards, and that grid will have to stretch out in some cases and shrink in others to get back to that group. And these are what are called Jacobian Determinants, and below Jacobian Determinants, as long as you take the exponential of them, will be 1 above or below 1. And above 1 would be when the grid has increased above 1, and below would be that. But you can take those Jacobian determinants in every voxel and compare them between the two groups and get a statistical significance between those groups and tell where something has increased or decreased in a given voxel that's 40 micron by 40 micron, 40 micron. So that allows you to look across the entire brain and see where the differences, the volume differences are happening. So again, we'd use regions for the first one, but because the thalamus was considered the same, it was a really big structure, small one. And rather than put any weightings in, instead of looking at these 182 structures we had, we looked at 64,000 voxels in every mouse that we looked at. To get to 64,000 voxels, we actually had to downsample the data from 40 micron to 200, which really makes my supervisors mad, because you go through all this work to get 40 micron and ruin it. But it makes it manageable, so I can actually do it at a reasonable time frame. And then what we do is we calculate the effect size differences between the 125 different models and their corresponding wild type. The effect size was calculated both in absolute volume, so millimeters cubed, as well as relative volume, so you get a sense of percentage of total brain volume in case there's like P10, which is just massive and everything will be big. And then this is just showing some examples of what these effect size maps look like. So an effect size is just how many standard deviations away the means are from one another. So an effect size of 1 means that the model group is 1 standard deviation higher than the wild type group. So anything shown in red, orange, yellow is big compared to its wild type counterpart, and anything shown in blue is smaller. And therefore you get these 3 different examples here showing you what these look like. And basically this is one of those 64,000 voxels, every one of these little pixels that you see here. And then from there, we use a technique which you may be familiar with. It's called similarity network fusion. Now, it was originally created to take 2 different data sets and that are different. So in the example they're showing here, you're taking the DNA methylation and mRNA methylation, both from the same patients. And what you do there is you create a similarity matrix, and you to compare the patients across one another, and then you're basically, from that, you create a network where the nodes are the patients and edges are similarity, and then you start to iteratively fuse that together to create this new network, which takes into account both the DNA and the RNA information. What we do is we take the absolute volume information and we take the relative volume information and we put it together to fuse that data set so that we have both the information from P10 being 10 times larger brain, but also individual structural variation within that rather than just bigger than smaller. So that's what we do to create our new data set, which has both the absolute and relative volume within it. Now,
why not analyze those? I mean, we use this method actually for integrating genomic data. It's a good method. Why not do that separately? Because I mean, they're asking kind of 2 different questions. I mean, they can be a related question, but it's kind of 2 different things, right?
Yeah. So the question was why add those together instead of doing them separately? The reason, to make it easier to analyze at this point, I find in the original paper we only used relative volume, and I felt like we were missing an aspect of the data. And then so we wanted to look at absolute volume, but when you do that, you get a group that's big, and you get a group that's small, and you get a group that doesn't change. And so adding the relative volume information into that tended to give you a better story. It refines that story, because that's not to say that P10 and other models that are really big don't still group together, but you get a little more variation in your clusters. So using the fused data set from this SNF function and correlation, we also didn't want to just pick an arbitrary number of clusters to cluster them in. So those that do cluster analysis, one of the biggest debates is how many clusters do I actually choose for this data set. And we wanted to kind of take that out of the equation, and so we did the data at 2 clusters, 3 clusters, 4 clusters, all the way up to 10. And then started to look at the consistency of models that would always go together, regardless of how many clusters there were, to get an aspect on really, really strong connections between models. And again, this allowed us to investigate the cluster in itself, and additionally take those clusters and start to link molecular pathways to them. So if we had 2 different clusters, and then we take all the mice in that cluster and look at the effect size difference for the cluster of, in this case, if we have 125 models, there's probably about 60 plus per group. You take the effect size of all those 60 and you take the median effect size and look at what that looks like for each cluster. At 2 clusters, you don't really see very much at this effect size threshold. They're too variable, you're not really getting consistent story. But then you start to look at 3, and then all of a sudden you start to see some patterns emerge in the cluster. So all of a sudden you have subcortical structures here in the midbrain that are increased in size, and still you're not getting a story from those other ones. But you 4, and then you can start to see, again, that group tends to stay the same, and this group starts to be an increased cortex with decreased white matter structures within, and maybe some decreases in the cerebellum. You can also keep going all the way to 7, and then you can start to see, well, maybe this is your P10 group down here with all this really large brain, but you also have that cortex, white matter, cerebellum thing. You have another one that's showing decreases, and then you can go all the way to 10 and look at that, and that's sort of what our 10 clusters would look like. And that is the neuroanatomical signatures for if we went all the way to 10 clusters. But the real question is, okay, with all of that said, what does it mean? And is it consistent? Does it make sense? So one thing I like to look at is can we confirm that the clusters are doing what we think they should be doing? And we had 3 different models from 3 different labs on 2 different backgrounds, created genetically. One was CRISPR-Cas9 and one was the normal way that you do it, which I forget the name of. Regardless, we had 3 different models of CHD8 that came from these 3 labs, and they always clustered together. So it is working. It does make sense. We also had 3 different models of 16p11 that were deficient in that copy number variation, as well as duplication that always went together. We had 2 different models from 2 different labs of KCDD13, which always went together. And then there are also some models that are both in the same syndrome. So in this case, it's Sanfilippo syndrome, which again always went together. So it made sense that our clusters were doing what we expected. Now, this is showing you some of the consistent connections. So there are showing you from like 6 different groups here, and these ones tended to go together in about 8 of the 9 clustering separations. So you get things like chromatin remodelers, ARID1B and CHD8 always tend to go together. You get FMR1 on 2 different backgrounds mostly going together, and you get Neurexin1 and Neuroligin1 going together, and these are genes that actually like handshake across the synapse. So it makes sense. You get your 2 behavioral mutants that are just socially behavior, and they tend to go together. You get P10, NHS also is large brain. It's Horn syndrome, and so the fact that they go together does make sense. This is probably our large group. But they also bring in 16p11 with a deficiency, which just has an increase in the subcortical structures, nothing really on the cortex. And tau K2 is actually within that CNV.
And, which is so interesting, because we did anyway, we can talk about it. Okay. We did a co-expression study and showed that was the gene that most correlated with the with the CNV.
Yes, and also, sorry, the question or the comment was about how tau K2 and the gene expression correlated with 16p11. But also, it seems like, so we've done quite a few of the models within 16p, and tau K2 actually is the one that shows basically the exact same volume differences. It seems to be also the volume in the CNV as well. Which makes sense based on the gene expression as well. And then one interesting one, we used androgen receptor mice as well, and they separated, you can see it, it's this group, oh wait, it's this guy here, and we can follow them, but basically they separate early and stay by themselves the whole time. Nothing really tends to go with them. So regardless of all that, where do we go from here? We have all this information of clusters. What can we say about these groups? Well, one thing we wanted to test was what did these clusters mean? And the Simons Foundation Autism Research Initiative, SAFARI, creates these gene scores. Again, this has been modified, so it's not updated because they no longer have a 4 and a 5. Because minimal evidence and hypothesized but untested were their categories. So they've actually removed that, so they tend to go 1 to 3 now. But the point of the next slide will still be evident even with that. So we wanted to see maybe, well, are all the category 1 genes in one cluster? Are all the category 2 in another cluster? Is that what's possibly driving these? And that's not the case. So what you're seeing here is your gene score is listed. 6 just means it's not listed on Safari database. But 1 strong red is the high confidence genes. And then as you get less and less red, there's less and less confidence. And so you're not seeing, and then each individual group box rectangle here is highlighted by the enrichment of those scores in that given cluster. So the darker the red cluster, the more high confidence genes are in it. But you see that there's not really a dark red cluster everywhere, and there seems to be a bunch of genes that are high confidence, again, being distributed amongst the different clusters. So it's not confidence level in the autistic gene that's clustering or causing the same neuroanatomical changes. Similarly, if you look at syndromic or not, whether it is Fragile X, whether it is Rett syndrome, and it is syndromic for autism, there's not a syndromic group either. So this is just a yes or no. Basically, it's red, it's a syndromic gene. If it's not red, it's not. And so there's not a category that is necessarily syndromic. This is about as close as it gets, that strong red. But again, it sort of disappears at the next grouping. So, and again, these are, the rectangles are the enrichment for syndromic at this point. So it's not gene score, it's not confidence in the gene, and it's not whether or not they're syndromic, so that's not what's driving these clusters. So the question we wanted to ask is, can we link molecular pathways to these clusters, and is that something we can do? Now, unfortunately, I don't have the new data for the 125 models, so you'll have to look at some of my old stuff. But the story is the same. You'll just have to trust me on that. So we used 92 for this study instead of the 125. And basically what we did to calculate the enrichment of these pathways, because in the cluster, when you cluster into 10, you're looking at about 12 mice per group, 12 genes per cluster. And then maybe you might have 1 gene in a given pathway in a given section. So what we wanted to do was we wanted to determine cluster neighborhood. So we looked for 1-hop interactions and included those genes into that given cluster to get a neighborhood that we could use. So a cluster neighborhood, as we called it. But first we had to take these 92 models, and because we didn't know what to do with CNVs, we just took them out so that we didn't have one CNV with 30 genes driving the entire story. And then for those that had hets and homozygotes, we just combined them. So in the end, we had 51 genes that we were looking at, and we used a string database to get the interaction information from that. And this was done with help from a graduate student in our lab named Johan. And so the goal is to get the gene neighborhoods for all the genes, all the 51 genes that we have. So we do a string database bioinformatics API call to basically calculate with a really high confidence the one-hop interactions for the given 51 genes. And so there was 919 total interactions with these 51 genes at that confidence level, which led to 749 neighborhood genes for the entire group. And that, depending on what cluster you're in and how that was divided up, you're looking now at, instead of 125 divided by 10, you're looking at about 70 genes per group kind of thing. Yes?
Is that network significant?
The network we're seeing here?
Yeah.
It's just showing the clustering, or the ones that are in, do you mean actually significant in statistics, or do I feel like it's a significant network?
So do 51 genes have edges more than expected by chance?
Yes, that is a significant network, yes. So now all of a sudden we have 700 and, well, actually 800 genes that we are now looking at in this group. And so we use the KEGG database, which is uses the molecular signatures database to talk about molecular pathways. And so genes have been annotated to these pathways. And we picked a priori 20 pathways that we thought were autism relevant, and then test the hypothesis that models clustered by neuroanatomy might associate with one of those clusters, or one of those pathways. So if we were to look at the MAPK signaling pathway, what you're looking at here is the brighter red, the more enriched for that pathway that cluster is. So the stronger red means there's more MAPK genes in this cluster neighborhood. So you do start to see a flow and a consistency for some of these groups that maybe there is a MAPK signaling pathway that's telling part of that story. You can also look at the Wnt signaling pathway, and you can see that there is a couple clusters that are significantly enriched for Wnt signaling. And focal adhesion, which is also a common finding in autism. You can also start to look at some deviation and separation in focal adhesion in a few groups here, and you can start to say, okay, these clusters might have something to do with that. This is significantly interesting. And so what we did was we took these clusters and the enrichment data that we had, and we did some pairwise comparison between the different groups. So for each pathway selected, we had a number of cluster neighborhood genes that were in that, and we wanted to see how many of those overlapped with a given pathway. So the little b is genes in our cluster neighborhood that are in the pathway, and big capital B is the total number of genes in that pathway that are annotated. And so if we had cluster 1 where there are 12 genes within the pathway, and cluster 4 where there's 5 genes in the pathway, we tested the proportion difference, measured a p-value, and also FDR adjusted that p-value. So in the case where cluster 1 had 12 and cluster 4 had 5, that wasn't a significant difference between those two clusters showing. That's okay. And then with all that said, we then, on this chart, we show anything that's listed on here in a given cluster is significantly different than all other clusters in that level. So when you're looking at, so we have a MAPK, adhesion, hedgehog, Wnt, long-term potentiation group versus an mTOR transcription factor group here at the beginning. But then as you see, as time goes on with the cluster deviation, you start to see a Wnt hedgehog pathway coming out at 6 clusters all the way to 10, or you have a MAPK adhesion one that's showing up as well. So there seem to be 2 clusters in the end that are associated with those pathways that seem to be consistent regardless, not irregardless, but regardless of the different genes that are listed there. So maybe then our story, instead of 5 or 6 different neurodevelopmental disorders, maybe we have a WNT section, maybe we have an adhesion or a MAPK group. But there's obviously more work to be done here. We've just started linking pathways. We also have this set up to look at Go terms. The problem with Go terms is we are going to have to curate the list, or you're going to be dominated by cancer genes or cancer stuff that shows up rather than what you're looking at. So we're going to have to look at a curated Go term lists. You also have disease terms. You have the Reactomics database, and all of these different things. You can, if we can start to tell the story a little better. There's also the aspect of, we're just talking about neuroanatomy here. You can use the similarity network fusion and add in behavior, historical behavior into the mix. You can add in gene expression because, as Dan was asking before, we have all the Allen brain information. So you can start to bring in all that other information to use more than just neuroanatomy to hopefully streamline this a little better. But with all that said, in conclusion, are signatures for autism in the brain with their neuroanatomy. There is groups, there is signatures that we're finding. And again, not one mutation is gonna define all of what autism is, and all these different signatures tend to have different mutations associated with it. But both human and the mouse data is suggesting that there is more than one autism. But I don't think that it's going to be one per identified gene. And I think it's also going to be mixed with these other neurodevelopmental disorders. And I think identifying the autisms will probably lead to better treatment, because if you can streamline the diagnosis, there's going to be better treatment outcomes. People are going to do better on their clinical trials, and that. So getting a better group to begin with is going to help the story. And I just want to acknowledge my 2 mentors, Mark Henkelman, who has retired and is probably, at least in the summer, sitting out on his deck at his cottage, and Jason, who is actually still involved at the Mouse Imaging Center, but has taken a position at the University of Oxford to run their mouse facility. So he does have an adjunct appointment, and still is getting me to run his lab in Toronto. So then, just Star Wars is always seemed to be in the theaters, and that work like this takes a lot of people. I can't fit it onto a slide, so we'll have to do it in a way like this to show everyone that's involved, because I can do this without the collaborations and people offering their mice and working with us and that. So it is very helpful. Anyway, thank you.